Hi, everyone. I'm Dr. Kara Wells, and I am eager to share our study entitled Analysis of Real-Time Embryo Morphokinetics in Relationship to Embryo Sex. The late George Seidel often said, sex is the most important economic and genetic trait, but we cannot select for sex through genetic selection. In nature, the sex ratio is very evenly distributed, but economically, people do care about selecting for embryo sex and shifting the ratio to either favor females or males. For example, dairies may want females for replacements as only females can lactate and produce milk, or they want male beef calves due to their rate of growth. Either way, there's typically a preference. However, controlling for the sex ratio is limited in scope. The most common method to control the sex ratio is by use of flow cytometry. This method involves dyeing sperm cells with fluorescent dye and passing them through a flow cytometer. Cells can be either sorted based on the variance of an X or Y chromosome, or the undesirable cells may be laser ablated. This method is highly accurate and widespread in use. Unfortunately, though, sex semen is known to produce fewer embryos and may reduce embryo competency. At the embryo level, preimplantation genetic testing may be used to determine embryo sex. This method involves a biopsy of cells, typically from the trophectoderm, which are sent off to a lab for genetic testing. The remaining embryo is frozen until results are received. This method is expensive, requiring hundreds of thousands of dollars in equipment cost, labor-intensive, requiring highly skilled embryologists, and requires several days to receive results. Additionally, the biopsy procedure poses risk to the embryo and may compromise embryo viability. Therefore, use of PGT, or preimplantation genetic testing, is limited. New developments using an analysis of embryo metabolites to evaluate sex are ongoing. These methods are usually involve sampling of the media surrounding the embryo, which requires a high degree of precision and control. These methods are promising as they are non-invasive, but still require expensive equipment to run the results. To date, these methods are not commercially available and are typically regarded as still being experimental. Therefore, our team decided to take a different approach to predict and identify embryo sex. In recent years, there have been major advancements in camera and computing technologies. We suspected that cameras could capture growth and developmental characteristics that are not perceptible to the human eye and brain when examining the embryos under a microscope. Our initial studies aimed to predict embryo viability. In these studies, we successfully developed processes and models to do this by evaluating embryo morphokinetic activity in real time. These processes allowed us to quantify embryo change, which we believe are a function of embryo metabolism. The scientific literature has many studies demonstrating embryo metabolism being sex-dependent. There are also many studies that demonstrate that male embryos grow faster and have a higher cell count than female embryos. Therefore, objectives of this study aim to use embryo video capture and graphic image processing to allow non-invasive identification of embryo sex. Our first approach used a form of graphic image processing called background subtraction to quantify embryo change frame by frame. In the top, there are two consecutive images of 6 embryos. In my opinion, they look exactly the same. However, there are slight changes. If we were to superimpose these images, they appear blurred. We can also use background subtraction, which subtracts the images from each other and highlights any pixels that are different. We can see the pixels that have changed. So here's the negative, and here are the pixels highlighted. We can now use a computer to count the changed pixels and quantify embryo change in real time. Let's take a look. In the raw video on the left, the embryos appear to be static groups of cells. But with background subtraction, we can see the embryo's real-time activity frame by frame. Other procedures allow us to see the direction and magnitude of the cellular activity. 2 years ago, we did our first sex selection study. This involved 102 embryos recorded on a 12-year-old Nikon Coolpix camera. We calculated the mean pixel change or morphokinetic activity of embryos that were known to produce either a bull or heifer calf. We realized, based on the mean pixel change, the male and female embryos tended to cluster together. We then used the k-nearest neighbors equation, which in our case compares the embryos with an unknown sex identity with embryo mean pixel change of known sex. We use the k-nearest neighbors equation to see how the value compares to the embryo values of the known sex. We found that if we take the three closest values, we were 61% accurate in predicting embryo sex. This study was promising, but it only tested 102 embryos. I also want to point out that the image quality of the 12-year-old camera was not great. We suspected a larger sample size and better camera equipment could improve prediction accuracy using the KNN or k-nearest neighbors analysis. Recently, we recorded a 30-second video of 173 fresh IVF embryos. These embryos were transferred into eligible recipients and fetal sex was determined by ultrasound. We then applied background subtraction to these embryos and calculated mean pixel change or morphokinetic activity. We found that male embryos have higher morphokinetic activity than female embryos. This finding was statistically significant. This was suspected as male embryos are known to have faster rate of growth and higher cell count than embryos of the same age. We also found that males have higher baseline activity. This was determined by calculating the sum of embryos' activity over the 30-second video duration. We then wanted to look at the influence of semen type on embryo activity. So we compared conventional semen that produced pregnancies or did not produce pregnancies, fresh sorted semen that produced pregnancies or did not produce pregnancies, and reverse sorted semen that either produced pregnancies or did not produce pregnancies. We found that the fresh sorted semen that did not produce pregnancies was statistically different than the conventional semen and had high variability. We also saw that the reverse sorted semen had the highest amount of morphokinetic activity and a great range and variability of morphokinetic activity as compared to the other semen types. This data allowed us to conclude that semen type does have an influence on embryo metabolism and can influence the embryo's cellular activity. Last, we want to repeat the k-nearest neighbors analysis on videos recorded on newer equipment. These videos were recorded on a 4K view microscope camera mounted into a trinocular port. The embryos were ranked from slowest to fastest. The light green represents the females, whereas the dark green represents the males. We found that embryo sex did tend to cluster together, and when the KNN or k-nearest neighbors analysis was repeated, we had a 74% prediction accuracy in correctly predicting the correct embryo sex. All in all, we learned that key indicators of embryo sex may be detectable in video image data. Our studies were conclusive in finding that male embryos have more morphokinetic activity than female embryos, and we are now working to do further studies to perform more advanced machine learning analysis to further identify patterns using convolutional neural networks that can be associated with either male or female embryos. I'd like to thank my team, Dr. Russell Killingsworth, Cameron Hayden, Dr. Joe Webb, Darren Dunlap, Michael Ray, Kevin Johansson, and Justin Wells for collecting, analyzing, and processing this data. Thank you.